My area of research is neurovascular physiology. You might describe that as plumbing meets neurology. The brain, what we call the brain parenchyma, is actually a very soft, squidgy mass of jelly. It's made up of neurons and fine blood vessels. And this is very delicate. And it's protected by the cranium, which is a hard, bony container. But unfortunately, if you were to allow the brain just to rattle around in there, it would get damaged. It would hit the sides of the cranium and would be very badly damaged. So actually it floats in a sea of water. And that water is, we call it cerebrospinal fluid because it has other components in it. And that cerebrospinal fluid protects the brain parenchyma from trauma. But it does something else. It also regulates a very complex biomechanical system that regulates the blood flow through the brain parenchyma, keeping it smooth and non-pulsatile. Really the aim of the research, I suppose the bottom line, was to better understand what is going on in the cranium from the fluid mechanics point of view. We know that the Windkessel mechanism is very important in regulating the blood flow through the brain parenchyma, and that this changes as patients age. So it might be involved in diseases such as vascular dementia. We worked on this project with a team in Milan with Dr. Marcella Lagana, who's a medical physicist there. And she did all the MRI work on, I was going to say patients, but these weren't patients, these were healthy young individuals, so they were subjects. And she collected the data using MRI through the neck to get the flows of the blood flows, the arterial blood flow into the cranium and the venous blood flows out of the cranium, and also the cerebrospinal fluid pulsing backwards and forwards in the neck. She also collected data in the cranium for the cerebrospinal fluid in what we call the aqueductus sylvius. And that actually is the flow into the ventricles. And the ventricles are a big space in the middle of the brain, which is filled with cerebrospinal fluid, which pulses. And it's never been fully understood how that mechanism works. And one of the things about the paper is that we've been able to explain this. It was really important to understand how the intracranial biomechanics, fluid biomechanics, behave in healthy individuals, because you can't understand or understand how this might influence pathologies, neurological disease, until you first understand what's going on in healthy individuals. And one of the problems is that while we can get MRI signals to measure the flow rates of blood and cerebrospinal fluid in the neck, and many research teams around the world have done that over the years, interpreting the signals that are produced are very difficult to interpret, and so it's very difficult to interpret what's going on inside the cranium from what's going on in the neck, and we've been able to do that. And so we believe that this is an important finding that will hopefully help future neurological research. It gives us a deep insight into how healthy individuals behave, how the fluid mechanics of the cranial space behave. And it opens up new possibilities to examine blood flows and cerebrospinal flows in the neck using MRI in patients with neurological disease, maybe with Alzheimer's, with multiple sclerosis, Parkinson's, or even suffering from migraine. And we might find that there are differences, changes going on in these flows, which we can then interpret as changes going on in the cranium. And so we're continuing to work with a team in Milan to analyse now patients who've got neurological conditions to see how the flows in the neck and the intracranial volumes change in these patient groups. So that's the work that we're proposing to do next.